Good morning, everybody. And good afternoon to the people across the ocean. Okay, so thank you, Olaf, and thank you, Eduardo, for the title of Italian Godmother to Roya. This is definitely the best title I've ever earned in my career. And I would like in the next 10 minutes to talk about the journey that we have embarked in. So when we first met Roya and Leila and Eduardo in the clinic, Olaf and I sat down and started thinking about the type of things that we can do to help. And for me, being in muscle biology, the tissue that I know the most is obviously skeletal muscle. And so I wanted to put my expertise to a good use for this, for Kabuki syndrome, putting together some of the resources that I already have in my lab. And so, as you all know, KMT2D and KDM6A are genes that control or regulate many tissues, including the skeletal muscle. And children with Kabuki syndrome show various degrees of muscle weakness. And so they become tired sometimes, they cannot endure. And so my team and my lab decided to approach the question whether the skeletal muscle tissue itself, including muscle stem cells or muscle fibers, which form this tissue, have a problem or in differentiation or proliferation that can cause this weakness. And so we decided to approach this work, by studying the differentiation and the regenerative potential of muscle stem cells in mice, as well as in humans. And I'm going to take you through this journey in the next 10 minutes or so. So when we first isolated through the Roya Kabuki program, the muscle stem cells from mice that are control or wild type, and compared them to the ones that have a mutation of a single allele in the KMT2D gene. And this model represents basically what happens in the patients as well. So the KMT2D gene is actually mutated in every single tissue. And so we can use this model as something that we can study to then project what happens to human. So what we did, we isolated the muscle stem cells that you see in these two pictures, and we cultured them in vitro. This is something that we routinely do. And then we use marker for stem cells to understand whether these stem cells remained inactive and therefore they were not proliferating or differentiating, or became more active with a mutation in KMT2D gene. And again, this is work that has been done in mice. And so what we saw, we cultured these stem cells for about 48 hours. And then we tested for markers that would allow us to understand whether they were remaining quiescent or stem cells or proliferating. And we saw no difference in these stem cells in the number of quiescent cells or the activated cells, suggesting that there was absolutely no problem in this part of the skeletal muscle component. And so the next portion of the study was to understand whether there was a problem in muscle stem cell differentiation. And so in order to do this, we were able to actually follow the differentiation track of muscle stem cells. So we have markers in skeletal muscle that help us exactly position where skeletal muscle stem cells are during the formation of skeletal muscle. And you see these markers that I have highlighted right here in red. MyoD is the first marker that basically can make another cell type, such as a fibroblast, turn into skeletal muscle. It's called a master regulator. And then followed by MyoD, there is myogenin. There is the first marker that drives differentiation of muscle stem cells. And then myosin, you all know, is a major protein component of the skeletal muscle. And so we follow these three markers during the differentiation of skeletal muscle cells from control mice on the left panel, and in mice that had one gene mutated in KMT2D. And so what we saw is that early differentiation and commitment to skeletal muscle, as labeled by MyoD right here, was not affected. So there was definitely the muscle stem cells could commit to become mature muscle in the KMT2D mice. and with absolutely no problem and no difference compared to controls. But when we were pushing the differentiation towards myogenic and myosin, where skeletal muscle becomes more robust and develops the contractile properties and forms muscle fiber, we started seeing some delays. And you can see right here in panels D and E how the myogenic expression was actually significantly reduced, and it was quantified in this graph, was significantly reduced in KMT2D mice cultures compared to control. And this actually trickled down to expression of myosin heavy chain, which is basically one of the major contractile proteins in the muscle fibers. And you see again a difference in differentiation and a significant delay. So what did all of this mean? And is this something that we also see in vivo in the mice? And more importantly, is this something that is irreversible? Because when we all think about therapy, such as the elegant approach that Dr. Bodamir just described in his presentation, we want to know that something that is already developed and in a KMT2D environment, for example, can be reversed. And we want to have some assurance that the drugs, will be effective in a formed animal model, or, hopefully soon in a human being. And so what we did, if we actually study the whole muscle of KMT2D mice, compared to control mice, and we want to see if there is a delay in regeneration in vivo, we can actually see this in the present slide. So what happens, we can actually injure the muscle of mice and then put the stem cells to activate. And as you can see here, we have a lot of myofibers that are stained in green. All of those green circles are muscle fibers that are undergoing regeneration. And then in red, you see the circles of laminin. This is an outline of the muscle fibers. And we can actually measure the size of these myofibers. And so these fibers, when we measure the size, we see that in KMT2D animals, they are significantly reduced in size. So what we had seen was that both in the cultures that I had just shown you, and as well as in the live mice, in the skeletal muscle fibers, have a reduced or slow differentiation capacity. And so our next question was to actually understand, as I was starting to mention, whether these stem cells could actually be rescued and the differentiation capacity rescued following introduction of these stem cells in a different environment that is not a KMT2D environment. So to approach this strategy, we used some resources that we already have in the lab. And what we did, we used control cells from mice that are unaffected and compare them to KMT2D cells that were extracted from the mice. And what we did, these cells were injected in a mouse that has muscular dystrophy, which is another disease that I have studied in most of my careers. And we injected the same number of cells in independent mice. And then following more than 2 months that we transplanted these cells, we tested for engraftment potential of the cells that have the KMT2D mutation versus the wild type cells. And we wanted to see whether in this environment that does not have a KMT2D mutation, but has a mutation in another gene that is called dystrophin, whether the cells with the KMT mutation had the same capacity to engraft and produce dystrophin such as wild type cells. And we were very pleased to see actually that the KMT2D cells were actually engrafting in the same capacity as well as control cells. And so this to us was extremely encouraging because it speaks about the potential of cells that have lived in a KMT2D environment to be able to overcome whatever delay in differentiation or other differences. They are able to overcome this and behave like a control cell. And so to us, this was a very positive sign and speaks to the possibility that perhaps also in patients, hypotonia can be reversed and some of the downstream effects of the KMT2D variations, may be overcome. So I talked about mice up until now, but what about humans? Obviously, through the Roya Kabuki program, we were able to, through the generous donation of some portions of skeletal muscle of children that were undergoing different surgical procedures, to acquire some discarded skeletal muscle tissue. And we were able to actually learn what happens in human samples compared to what we saw in mouse samples. And as we often find in the lab, what we see in mice does not exactly represent what we see in humans. And it speaks for the importance of doing studies on both systems. And so right here on the left side, you see a control tissue where the muscle fibers are in pink, and you see how they're nicely packed with one another. They are basically contractile units, and these are the ones that make us move. And all the examples in the next panels are from muscle biopsies from patients with a KMT2D variation. And so you can see that there are some differences, and some of the most prominent differences that we see consistently in all of the human samples is presence of these infiltrations, which we call fibrotic infiltration. And so we have seen that there is increased fibrosis, and this is something that in the mice we have not seen at all. So right now, we have analyzed only a handful of patients, about 4 or 5, and we're currently recruiting for additional samples because we want to understand the root of the cause of this fibrosis infiltration in human samples. There are fibroadipogenic progenitors in the muscle, and that are present in both human and mouse muscle that really have a crosstalk in the muscle with muscle stem cells. And we want to understand whether these fibroadipogenic progenitors, particularly in human samples, are somehow overactivated in humans and not in the mice, or whether this crosstalk between muscle stem cells and fibroblasts in the muscle is somehow altered in human muscle compared to mouse muscles. And so we are trying to recruit and understand whether the fibrosis changes with age, if it becomes worse, or whether there is a different biological problem, as I said, with intertalk between muscle and fibroblast. So in conclusion, I have shown you that muscle cell differentiation is decreased in vitro and in vitro in KMT2D mice. But the promising result was that when the mice were challenged, the cells were challenged in a non-Kabuki environment, they were actually completely rescued. And so this leaves the open possibility that hypotonia in Kabuki syndrome could be corrected when KMT2D is actually rescued. The mouse models show similarities to the human tissue, but also show some difference. And the fibrosis is something that I have pointed out to you a few minutes ago. And so we are committed to continue our work on both mouse and human models, and to use the two systems to actually understand exactly what is happening and how we can intervene in both models to correct any downstream deficiency. We want to study also the connection between muscle and nerve. We have done some preliminary analysis, and we recently published that data. It's actually open access in this month in the FASEB journal, and it's open to everyone who wants to read it. And then we want to deepen this understanding to see if the firing of the nerve into the muscle is similar in KMT2D versus control mice. And then we want to understand why hypotonia occurs in Kabuki syndrome. And again, working with humans is a very different system than mice, and we need to bulk up our tools for that. But clearly we have strong support and invaluable resources through the Roya Kabuki program about this. Last but not least, I want to acknowledge all the people who have worked with us in the lab. The member of my team are listed here. Alec Wright is actually a person that I would like to deeply acknowledge for taking during the pandemic some of this work and really bring it to a publishable level. Of course, none of this work would have been possible without the Roya Kabuki program team, Olaf Bodemer, Tala Deri, and Christina Hang, who helped us not only with the human samples, but also with conversations about intellectual conversations on how to approach the problems. We had colleagues in Cincinnati Children's Hospital that we have worked with, and they done some initial studies on the mice, and that's how we connected with them. So thank you to Andrew Lindsley's team when he was at Cincinnati's. And then, of course, last but not least, none of this work could have been done, especially in human work, with the generous contribution of patients and families who were willing to donate muscle for research when kids were undergoing other procedures, and they were willing to donate any discarded material. And again, none of this work would have been possible without the support. The research is expensive, and the Roya Kabuki program has made it all possible. So a big thank you to Eduardo, Leila, and their families for their support over the years and for everything they have done for Kabuki syndrome.